Welcome to iExperts University. I'm Dr. Eli Sluch, and today we'll be talking about clinical genetics. In this lecture, we're going to learn about the terminology and the concepts of clinical genetics, the different chromosome analysis and tests that are available to test for the gene defects. We'll talk about the patterns of inheritance and what that actually means clinically and to people, and a little bit about counseling and management. Let's learn a little bit about terminology. So, hereditary means results directly from an individual's gene composition and can be passed down to subsequent generations. Genetic specifically refers to a defect in a specific gene. Familial means occurs in more than one family member. So, if it's just one person, it's not familial because it's just one person still. And congenital means present at birth. Genetic heterogeneity is a concept that the same phenotype can be caused by two different gene mutations. There's three different subtypes. So locus heterogeneity is the same phenotype is caused by two different genes. For example, Axenfeld-Rieger syndrome is caused by a mutation either in the FOXC1 or the PIDX2 gene, which are completely different. There is allelic heterogeneity, which means that different alleles on the same locus produce abnormal phenotypes. And then there's clinical heterogeneity, where different mutations at the same locus produce different phenotypes. So we can see that in RP, we have RP, which is retinitis pigmentosa, but it can be autosomal dominant. It can be autosomal recessive. It can be X-linked, and it can be mitochondrial too, just because what we see clinically is retinitis pigmentosa, but in reality, the photoreceptors are dying and it's happening due to different reasons. So just because we see something as the same, does that mean the same gene is affected? Chromosomes are giant bundles of DNA. We have 23 pairs because we're human for a total of 46. And genes are the small specific loci located on the chromosome. DNA is structured in such a way that each gene is located in a specific region of each chromosome. So if the gene A, for instance, at the tip of the chromosome, it's going to be located there for all people, unless, of course, there was a genetic translocation and the gene shifted. An allele is a term for a gene pair. So, as we know, we have two pairs of each chromosome. So, there's a copy of a gene on each one of those, except for XY combos. So, if you have a homozygote, that means that each gene copy on each of the chromosomes is the same, versus a heterozygote means one of the genes is different from the other one. You can have polyallism where a gene can have different mutations, so there's multiple variations of what each allele could be. And you can also have compound heterozygotes where you have two different mutant alleles at the same locus. Now, if we remember back to middle school biology, there's meiosis and mitosis. So, meiosis is the process of making gametes, versus mitosis is regular cell division where a cell makes an identical daughter cell. Mitosis is used for growth and maintenance, and the number of chromosomes in mitosis stays the same. If we look at the diagram, as we remember, we have our chromosomes, we duplicate the chromosomes, then we separate the cells out so each one gets an identical pair, and now we have two identical daughter cells from where we. As I mentioned earlier, meiosis is the process of making gametes. So the gametes are haploid. They only have 23 chromosomes. They don't have a pair. The reason for that is when the sperm meets the egg, the haploids will merge to create a new cell. That's why the new cell has the same number of chromosomes as the original cells from which the gametes was derived. So we have the correct number of chromosomes afterwards. So each gamete represents only one pair, but because of random assortment, it's variable. So each gamete is unique in itself, which is very different from mitosis, where everything eventually is the same. The number of crossover of genes and alleles is like 50 per meiosis and 1 to 3 per chromosome. So there's a very large number of recombinations going on when the gamete cells are created. Segregation refers to the phase of meiosis where the genes are basically eventually segregated into each of the haploids. So the allelic genes, which are, remember, the two of the same gene on the pairs of chromosomes, start getting separated, and each one goes into a different gamete. Compound heterozygote cannot transmit both mutant alleles because one is normal and one isn't. So one of the gamete cells will inherit the mutated version of the allele, and the other one will inherit the normal one. When you have non-allelic genes on different chromosomes, they can also switch through gene translocation at the time. So just because we have the same number of chromosomes, if you look at the differences in the metaphase where they all line up against each other, you can see the alleles can start jumping from one chromosome to the other. So when you look at the diagrams just for color, you will see that the gametes don't wind up with just blue chromosomes, just red chromosomes. They are kind of striped, and that's because the genes will switch from one to the other due to the way meiosis works. What's interesting is you can have something called linked genes. So the further the two genes are from each other, the higher the chance that they can make this jump and recombine into this combo chromosome. But the closer they are, the probability decreases just because of the length between the genes. So when genes are linked, they are pretty close together, so they usually will not do this jump. Now that we understand the difference in mitosis, we can actually start looking at chromosomes and genes. So originally, genes were analyzed via karyotypes. So we take T cells in the metaphase stage where the chromosomes kind of nicely coil up into their shapes, and we arrest the cell division of colchicine. Then we can actually take each chromosome on a slide, stain them with fish hybridization, which are specific DNA fragments of a fluorescent marker, and they target specific genes and alleles on the chromosomes, so they kind of cause this stripy pattern. And based on this pattern, you can, one, match the chromosomes, because again, the alleles are pretty similar. And you can also arrange them in size so you can see which chromosome is which. I mean, this is a clinical drawing. In reality, it looks a little different, but you can still tell. Another way to look at genes, because karyotypes are good for large chromosome abnormalities, like missing chromosomes you can see in a karyotype. Chromosome tail that got chopped off, you can see that too. But if you want to look for specific gene mutations, we would use the chromosome microarray technology, where we have chromosomes lined up on the slide basically. We label sample DNA and we plate it on this reference plate. And then we can tell the relative intensity of the genes that got stuck and the ones that didn't. So you can see the signals of kind of red, green. And based on the signal intensity, you will be able to tell if the allele you were looking for is missing, or if that part of the chromosome is missing just because the signal intensity is not there. Lastly, you can do gene sequencing, which is what is the most popular and probably most cost-effective thing right now, where we take genes, sequence the entire genome, and then we specifically look at DNA base pair analysis and compare it to the known DNA libraries. And this way we can specifically identify if a mutation is present. Aneuploidy refers to the fact that the cell has an abnormal number of chromosomes. This does not count towards gametes because gametes are haploids. We know that already. So trisomy is 3 copies of our chromosome. So if we have an extra copy of a chromosome, we're going to express too many gene products, so the ratio will be off, so that can cause disease. Monosomy is we only have one pair of a chromosome, and it's abnormal because the number of genes that we need to produce is now cut in half. Therefore, it doesn't work well. The only exception to this is XY, and the bar body in XX. To have the correct number is actually 1. That's why the bar body deactivates one of the X chromosomes in females. So they become functionally X monosomy. But other chromosomes you need 2 of. Now, this happens for several reasons. One, you have meiotic non-disjunction, which during the gamete separation stage, if the chromosome pairs don't split up and both pairs go into one of the haploid cells, it has an extra chromosome pair and the other one is missing that chromosome entirely. So when it combines with a normal haploid, the ratio will be off. It will be the +1 or it's going to be missing 1. Sometimes you can have, again, lag where the chromosome just fails to migrate and just gets lost during division. But the idea in the end is if you have the wrong number, you can have disease. And depending on which chromosome you're missing, the disease severity will be different. So Down syndrome, trisomy 21, the chromosome is pretty small. So yes, you get an extra pair, but it turns out that it's not lethal versus most other aneuploidy conditions are lethal. X chromosomes do tend to be somewhat of an exception just because if you have an extra pair of X, there's some X chromosome deactivation through the bar body mechanism, so eventually it does get turned off. And if you're missing one of the X chromosomes, you can still become female, but it turns out you need some of that normal XX for normal development. That's why even Turner syndrome, which is monosomy X, those people are fairly normal, but they still do show some variation. Now we're going to talk about some specific syndromes specifically. So trisomy 21 or Down syndrome is the most common chromosomal aneuploidy syndrome. The incidence is about 1 in 800 live births. It tends to be more common with advanced maternal age and slightly elevated risk for very young mothers as well. The 95% of the time is the extra chromosome is from non-disjunction, as we talked about before, and sometimes it's from translocation, where the long arm of 21 actually attaches to another chromosome, usually 14 or 22. So when you actually count the chromosomes, you have the correct number, but 14 or 22 becomes very long. And this way you can actually, again, pass it down from generation to generation just because it's attached to that chromosome. The translocation, though, is not related to maternal age. It's just a non-disjunction. Pictured here is the actor Zach Gottsagen. He is the guy who has Down syndrome, and he was the actor portraying Down syndrome in the movie Peanut Butter Falcon. Clinical features of Down syndrome typically manifest with developmental delay, mental retardation, short stature, hypotony, in the flat occiput, which is the back of the head. There's some finger hypoplasia. The crease on the palm is all the way across the hand and not up and down as in normal human development, possibly due to reduced motility in the hypotony. Eye-wise, they have these brush field spots, which are little white spots in the iris. They can have strabismus, nystagmus, and are actually predisposed to keratoconus and cataracts. Back to terminology, let's talk about mosaicism. So mosaicism means an individual or tissue contains 2 or more cell lines with different chromosomal constitutions. So the mechanism is the mitotic non-disjunction now. So when we have our embryo, the gametes have formed a new line, and those cells now start replicating through mitosis. If one of those cells at an early stage develops chromosomal non-disjunction, the daughter cells will have different number of chromosomes, and then they will continue to duplicate themselves. So this way you can actually get an individual with some mosaicism downstream in their cell line. Mosaicism is a little difficult to predict because it depends on when it occurred during embryo development. So usually the cells of the normal chromosome number and the trisomy cells survive just because they have extra material. Uh, Trisomy 21 can have mosaicism too, where you can have Down syndrome features, but otherwise appear clinically normal. The number of trisomy cells in the blood may also not collaborate with severity because just because the blood cell line has trisomy 21 doesn't mean the other organ tissue will. Gonadal mosaicism occurs if the mosaicism event happened during gonad cell formation in the reproductive tissue. So that means your gametes are not all bad, but also not all good. So you have a much higher risk of passing the mutation because some of your cell lines have an abnormal amount and some don't. This is a calico cat. The calico cat is actually mosaic female, depending on which of the X chromosomes got deactivated because they have different colors. Mutations represent a change in structure or sequence of a gene. So as we talked about before, we can have point mutations where we substitute one nucleotide for another. That's the most common. But then we can also have gross mutations, which form deletions, translocations, inversions, the big stuff. Non-coding mutations may not have clinical consequences just because they don't code for the gene itself. They're more in the intron part. Versus if they are more in the exon, there's a higher chance that something bad will happen. New mutation frequency is about 1 per million per gene per generation. So considering how many genes we have, something is probably happening right now. Since the big theme for today's lecture is definitions, we're going to keep going. So what is a genome? A genome is the sum of the total genetic material in a cellular organism. So the human genome, the cat genome, we want to know what's basically composes human from a DNA standpoint. A genotype is the genetic constitution in regard to a specific locus or set of loci. So specifically, what kind of gene for vitamin C do you have? Do you have the normal one that's inactivated, or do you have one from another animal that is activated? Hopefully the normal one. A phenotype is the observable characteristics. So just because we see something abnormal or normal doesn't mean that we necessarily know what gene does that translocate to. As we talked about RP, on the right you see corneal verticillata, which can be related to Fabry's disease, but it can also be due to amiodarone toxicity. So just because we see verticillata doesn't necessarily mean that that gene is defective. We have to test for it. Now, a phenocopy is the environmental or the non-genetic cause that closely mimics the phenotype, hence the example of amiodarone. Variability, anticipation, penetrance. So variability refers to the fact that just because someone has the same mutant gene doesn't mean that they're affected to the same degree. So even in families, when different children have the same disease, they may not have the same severity, and that is likely due to other gene combinations either trying to help out and make it better and try to help compensate, or they have mutations of their own and also kind of make it worse. Anticipation is an interesting phenomenon where the disease manifests earlier and more severe with each successive generation. And this is thought to be due to trinucleotide tandem repeats expanding with each generation. So, example is Huntington's disease. That's Woody Guthrie on the right over here, a famous Oklahoman folk singer, and he had Huntington's disease, and his children got it more severe and earlier. So each generation just keeps getting worse because in Huntington's, the trinucleotide tandem repeat is just bigger and bigger, destabilizing the gene further. Now, penetrance refers to the all or nothing concept. So it turns out it's kind of similar to variability, is that in some diseases or individuals, if you have any of the gene, then things can be bad. But sometimes if you have, let's say, a heterozygote, there could be enough of the gene to actually compensate to not show the disease. So it all depends on the penetrance. If you have incomplete penetrance, even in autosomal dominant disease, it is possible that that individual may have had enough to compensate for having the disease symptoms. Thus, you get the kind of the apparent skip in a generation phenomenon where you think that you don't have the disease, you weren't tested, and then you passed it on and you still have it just because you got lucky and your genetic material was able to compensate for the deficiency. Expressivity and pleiotropism. So expressivity, it refers to the fact there's a variety of ways and severity levels in which a genetic trait can manifest in different individuals. So kind of like variability. So example, NF1, you can have a child that just has cafe au lait spots. You can have someone that just has Lish nodules, or you can have somebody with horrible neurofibromas. And it's all from the same gene. It's just between different people. Pleiotropism refers to the fact that a single gene mutation causes multiple phenotypic abnormalities. So again, you have Marfan syndrome, you have one gene, but it does a bunch of different things, so a lot of things go awry. So in Marfan's, you get lens dislocation, as you see in the picture on the right. You have normally long spider fingers. You can get aortic aneurysms, and you're very tall. What does it have to do with the lens? It is collagen, but you get interesting phenotypic pleiotropisms. Now, most genetic diseases occur without regard for race or ethnicity. But some do tend to be concentrated in specific groups due to historic circumstances. So we have Tay-Sachs, which is more common in Ashkenazi Jewish ancestry, with a much higher rate of 130 versus 1 to 300 in the non-Jewish Americans, just due to population concentrations in the past. And we'll have a Gucci disease, which is predominantly in the Japanese population, and sickle cell in Black African ancestry. Recessive versus dominant genes. So dominant gene is expressed whether it's by itself or if it has two copies of homozygote. Just one copy is sufficient. A recessive gene, on the other hand, only is expressed as a phenotype if the copies are the same in a homozygote. If there's a normal allele, it's going to compensate for the recessive gene. Sometimes you can even have codominant expression where each of the alleles gets expressed, for instance, blood type. But usually it's recessive or dominant mechanism. So for recessive traits, usually it's an enzyme deficiency. So if you have one enzyme, the enzyme is good, so one copy is enough, and it can still make enough enzyme to compensate for the disease. Versus heterozygotes, even with the enzyme deficiency, do just fine and clinically unaffected. Dominant traits usually are like structural proteins, so the problem is they still get expressed, they get used, but the structure is bad, so things kind of fall apart. So fibrillin and Marfan, collagen and Stickler, causing cancer through proto-oncogenes. One bad copy is being made, and the fact that it's there, it's causing problems. Versus recessive genes, it's just something is missing, and a heterozygote can compensate for it. So Peter Dinklage has chondroplasia. And interesting thing about achondroplasia is the homozygous version is lethal, but if you're a heterozygote, it just causes dwarfism. Autosomal dominant inheritance. So by definition, the allele has to have two mutant copies. So the offspring will get a mutant copy of the gene. The question is, does the other parent pass on a good copy or a bad copy? So by math, if we have a bad copy, then the offspring will have the disease, and if they have the good copy, then they will be carriers. So traits typically appear only in siblings. Usually the ratio is again 1 to 3 if both parents are heterozygote, and only one person will get it. If one parent is normal and one is a heterozygote, then you will not see any issues. Now, typically, again, by math, we have the subsequent child of carriers, but about 25% affected, 50 carrier, 25% normal. That's again the square diagram on the right. So usually these are fairly rare because, again, to get the disease, you have to get two heterozygotes or one homozygote with disease, and the odds of meeting them are pretty low, unless, of course, you have those geographic concentrations that we talked about before. Autosomal recessive diseases tend to be enzyme defects and cause errors of metabolism. So we have alkaptonuria, we have albinism through the tyrosine kinase, we have a cute little albino penguin on the right. We don't know if that penguin has foveal hypoplasia and nystagmus and can't see, but the picture is pretty. And we can only find out if you're a carrier or not by doing gene testing because it's autosomal recessive and carriers stay hidden. Now, autosomal dominant disease is much more prominent in families. So we just need one affected copy and it will pass on to everybody. So just a single dose of the gene is enough to interfere with normal function. So usually you see this vertical transmission. So every generation you will have somebody as long as it's going to be passed on. And each affected individual has an affected parent. Unless, of course, this is a new mutation, because sometimes mutations are de novo and they don't just run in families. So all the families have to have gotten it from somewhere. Now, the important thing is autosomal diseases, if they're autosomal dominant, then if you didn't get it, you're not going to pass it on. Autosomal dominant diseases are usually structural non-enzymatic proteins because they still get built and things are just not working well with the product that they're making. Imagine building a Lego structure where half your Legos are gummy worms. That's not going to hold out very long. So typically homozygotes are much more severe and usually those are lethal. The variable expression is, again, more prominent here than in autosomal recessives just because in autosomal recessives tends to be all or nothing versus here sometimes they affect things more than others. Now you need to have conclusive evidence that this is autosomal dominant by having 3 more generations because otherwise it could still be random, or you can do gene testing. Gene testing solves all the problems and this is a known mutation. Now, interestingly, the de novo mutations are in elderly males. So if you're an elderly male thinking about having children, you could make a de novo mutation. Now, X-linked recessive mutations are very interesting because, as we know, males only have one X gene and females turn one of their X genes off. So males, by definition, are all hemizygous, and females typically are just fine because they have two, so they act as autosomal recessive from that standpoint. So usually, if you have an X-linked recessive disease, only the males are affected, and the daughters are usually all carriers. So what's interesting in this picture is this is the Russian royal family. This is the Romanovs. If you look at this picture, you have Tsar Nicholas in the center, and then he's surrounded by women and one son. Now, in this picture, the son has hemophilia because he got the X gene because his mom is a carrier, And the daughters over here, the other four, statistically speaking, half of them will be carriers of hemophilia. So Queen Victoria actually spread the X-linked hemophilia gene in many royal houses in Europe. The X-linked dominant disease is the analog to autosomal dominant, where one copy is causing disease. What happens is that females will be heterozygous with one bad gene, and they'll pass it on to either one of their children, boy or girl, at about a 50% rate because they have one chromosome that's good and one that's bad. Now, males typically will die from this because usually X-linked dominant is lethal in males. So despite this pedigree showing this female passing the gene on to her sons, those sons should actually be dead. Versus the female will have the autosomal trait and continue to pass it on. Now, just like the other autosomal dominant, if you didn't get the disease, you are not going to pass it on because you did not get the bad gene. Lionization is the hypothesis by Mary Lyons that one of the X chromosomes gets inactivated to balance out uh, the chromosomes to make the correct number of genes. Now, it appears that it's done randomly and forms this bar body, which happens about 6 to 11 days after fertilization. So, as we can tell, the cells have had quite a few minutes and hours to replicate. So, this happens in multiple cells. That's how females become mosaics. Now, polygenic and multifactorial inheritance. So it turns out that some traits or diseases are actually a collaboration of multiple genes, so they don't have the easy traditional genetic inheritance that we just talked about. Some of them can be multifactorial, some of them are environmental factors and combination of genetic factors. So most chronic diseases are multifactorial, which basically means things happen and we're not 100% sure why. So height, iris color, refractive error, even glaucoma are all different and polygenic and multifactorial, and there's usually not one gene. Like even the MYOC gene on glaucoma, it's associated with glaucoma, but it's not a really true genetic inheritance. Now, we talked about pedigrees today, but we didn't actually talk about how to read it. So, pedigree is this diagram which shows how genes flow through the family. So, the proband is the patient that brings the disease to our attention. We usually would indicate the proband by a pointing arrow. Women are indicated as circles and men are indicated as squares. If they have the mutation, we fill the square, the circle in. And if the circle and the square are connected, that means it's a mating pair. Now, the important thing to know about these is that sometimes families are not the best historians. There's also aborted fetuses, stillbirths, so the more information we can get, the better. Now, remember, if there's a consanguinity event, which is a brother-sister marriage or the kissing cousins, the chances of a genetic mutation really goes up, especially for those autosomal recessives, just because the probability of the event of them being the same are higher. And the other thing to keep in mind is that the national non-paternity rate is 5% in the US, 15 in an urban center. So just because someone thinks their dad is their dad does not necessarily mean that. Now, for genetic counseling, the goal of genetic counseling is to impart knowledge to the patient, come up with a diagnosis if we are able to do a gene type and identify the gene mutation, discuss the repercussions, and discuss reproductive options. The whole point of genetic counseling is it's non-directive. You should not be telling people what to do. You're just trying to explain information. So the options are you can accept the risk of children, you can delay childbearing, you can try in vitro fertilization. There's many options you can consider adoption. So there's many things you can do. The other thing is now we have prenatal testing. So while you're pregnant, you can get tested and see what genes or gene defects your child has to make the decision. Let's talk about some special topics of genetics now. So there's something called pharmacogenetics. So it turns out that we're familiar with genes that cause diseases, but we have genes for enzymes that metabolize drugs, and we have drugs that target enzymes. So it actually turns out that our genetics is important to how we respond to certain medications. As an example, isoniazid is inactivated by liver acetyltransferase. We can have an autosomal recessive low quality acetyltransferase where isoniazid will be toxic, or we can have a normal copy where isoniazid is just fine. So as we learn more about genetics and medications, we actually will be able to use genetic testing to predict how we respond to medications, choose better medications based on our genetic makeup, and avoid toxicities of certain medications if they're related to our genetic composition. As an example, in ophthalmology, G6PD deficiency is linked to hemolytic anemia, and depending on what medications we give them, we can cause certain problems. For instance, dapsone, you want to test people for G6PD deficiency because if they have it and you give them dapsone, you will cause anemia in that patient. And I have done that, even with genetic testing. Now, what can we do realistically if we identified a bad gene and we have a genetic disease? So, if it's a metabolic disease, we can do dietary control like PKU. You can avoid phenylalanine, and that's why many foods have a label that says contains phenylalanine, because if you avoid it, then you don't really have a problem. You can treat the excessive products, like in Wilson's disease for copper metabolism. You can give penicillamine that will wash out the extra copper. You can replace gene products that are missing, like in hemophilia or other coagulation disorders. You can sometimes give vitamins that kind of help sustain enzymatic function despite some of the proteins within the cascade not working. You can treat the symptoms, like in familial hypercholesterolemia, you can treat them with statins, or you can use gene therapy to replace genes or turn them off. As we talked about before, you can try to use the adenoviral vectors in already grown individuals, or you can target embryos with CRISPR to specifically cut out the bad genes.